Hello friends, in continuation to our yesterday's class, we shall discuss about DNA markers. DNA markers. Today we are going to discuss about two important markers, STRs, short tandem repeats, and SNPs, single nucleotide polymorphism. Short tandem repeats, otherwise called simple tandem repeats, they are important biomarkers, that means DNA markers. We have discussed in the yesterday's class what exactly the DNA markers and how they, that means their role in recombinant DNA technology. Simply they involve in gene mapping. Gene mapping is nothing but identifying the location of gene locus and also the distance between the genes. That means they also help in phylogenetic analysis and taxonomic issues. But the thing is, not accurate idea, but approximation of gene location is a crux point for DNA markers. And in the last class we have discussed about restriction fragment length polymorphisms and variable number tandem repeats, VNTRs. And in that line, the STRs is one more important. Short tandem, the name it implied, repetition is not for an extended length, but for a short period. Unlike VNTRs, which extends for example 10 to 100 base pairs. That means the size of the repeating unit is between 10 to 100 base pairs with respect to VNTRs, but here the size of the repeating unit is just 2 to 6 base pairs, and the number of copies is just around between 10 to 30. That means short repeating units, 10 to 30 copies, wherein the repeating unit is very important here. The quantum of repeating unit is between 2 to 6 base pairs, generally di to tetranucleotides. The rate is, for example, let's put a dinucleotides, 30,000 base pairs. This is very important. The rate of occurrence of short tandem repeats is 1 in at least 30,000 base pairs, and the number of copies is between 10 to 30, and the repeating unit between 2 to 6. In general, I mean usually di to tetranucleotides. Say for example dimer, for example GA. It is a repeating unit, and it may repeat for around 10 times in one organism, that means one individual. It may repeat for 5 times in another organism. I told you already, DNA markers are essential in gene mapping, and they are specific. They are different in different organisms. They are used to different polymorphisms. This is very important. But when compared to VNTRs, the quantum of short tandem repeats is relatively less, but they are more efficient than VNTRs. We also discussed about, let me have a note on comparative, okay? And as an illustration, how short tandem repeats will come into existence. There are different things connected with this topic, Say there are 2 DNA strands. So DNA 1 and one more. Say DNA 2. This is one organism and this is another organism. You magnify the same, it contains like GT is a dimer which is repeating for 5 times, 1, 2, 3, 4, 5. It's a dimer. Then the same thing in another, that means DNA 2. Have a different scenario. Magnify the same. it may repeat for 10 times. That means here the only difference between these two organisms is a repetition, number of repeating units. This is very important. Here is a dimer, it is also dimer, but the number of repeating units between the two individuals, say the DNA of 1 and DNA of 2 is different. That's the difference, and that is how the short tandem repeat, otherwise called microsatellites. STRs otherwise called microsatellites. Because of this point, they become crux point for DNA fingerprinting or DNA profiling. And their profile, that means here the repetition. Here the repetition of the repeating unit, that means with respect to number of repeating units, is different in different organisms. I may have a particular sequence, for example, I have the GT, that means a dimer, it is repeating it, it may repeat for 5 times in my DNA, but some other guy may have 10 times, some other may have 15 times, some other may have 20 times. As I told already, then copies may be around between 10 to 20. This is very important. So that means since the differences in the repeating units are high, they become unique. That is how they become crux point for DNA fingerprinting. And how they evolve. With respect to evolutionary perspective also we have to discuss. What makes the short tandem repeats come into existence? Because of mutation. What kind of mutation? This is called popularly known as DNA slippage. This is very important point. DNA slippage. It is otherwise called replication slippage. Single strand mispairing. During replication, we all knew that DNA has to denaturate, then copy. But here the thing is different. What we call DNA slippage. That means after denaturation, discontinuous. It's like displacement. Displacement of one strand. Since that means displacement of that particular strand leads to mismatch complementary pairing. This is very important. The strand which actually are supposed to participate in the complementary base pairing has been displaced. And in that position, a mismatch will come into existence. And because of this kind of DNA slippage, what we call replication slippage, the short tandem repeat, meaning STRs, otherwise called microsatellites, come into existence. This is a reason for STRs, and that is why they are the spots of high mutation rates. STRs are the regions of high mutation rates. I told already, whenever there is a mutation, that is a raw material for evolution. Since here the mutation rate is high, associated with DNA slippage, then it becomes, that means higher the mutation, higher it becomes the uniqueness. And higher it becomes uniqueness, and each individual will have their own STRs. And one more important point, within families, they may have unique STR sequences. That is why they play an important role in parental analysis. And the next important theme is how they differentiate and why are they best, that means better than VNTRs. This is a very important point. I think when compared to VNTRs, we have to discuss about these points also. Because latest DNA fingerprinting, has been depending on STRs. Initially, RFLPs, that restriction fragment polymorphism, and VNTRs, variable number tandem repeats, they used to have an important role, but now STRs have occupied the place of VNTRs and RFLPs in DNA fingerprinting. And what is the difference? They are short tandem repeats, otherwise called simple tandem repeats. Short tandem repeats. And here they are variable number tandem repeats. Tandem repeats. We're going to discuss the essential distinguishing features, okay? Short tandem repeats, they are evenly distributed throughout the genome. evenly distributed throughout the genome. This is a very important point. Why they are useful in current, that means latest DNA fingerprinting, is because they are evenly distributed. But VNTRs, otherwise called mini satellites, you know, mini satellites, they are restricted to the ends of genome, what we call telomeres. You know, telomeres. What exactly the telomeres do? They prevent the chromosome from the exonucleases active and also prevents from fusion with the neighboring chromosomes. But generally the VNT are restricted to ends, but it is homogeneous, evenly distributed along the length of the genome. For example, 1 in 30,000 base pair is a very important point. For example, 1 in here dimer, a dinucleotide repeat, for every 30,000 base pair. This is very, very important. And one more important, short tandem repeats originated because of mutations we have already discussed, DNA slippage. That means denaturation, displacement, mismatched complementary base pairs is the basis for the existence of short tandem repeat. And in respect to evolutionary perspective, also is very important. In evolution, that means how during the course of DNA evolution, it also becomes an answer. Why and how the DNA contains repeated sequences. How the repeated sequences came into existence. Here, the STRs become one of the signal, one of the what we call one of the indicators for that repeated sequences. They indicate in the way of DNA slippage. Because of DNA slippage, a kind of mutation that leads to STRs, and STRs is becoming an answer for why the DNA contains repeated sequences. This is a very important point. Repeated sequences and how they evolve. That means with respect to evolution perspective also, STRs are vital. They are vital in many spheres. The point it depicts. I told you already, I may have 5 repeating units with respect to GA, other may have 10, 15. On an average, they are unique. And they may have STR unique sequences may be present within a family. This is very important point. But VNTRs are different. They just have to telomere, that's it. And they depend on the differences in the repeated units, that's it. Number of repeat, that's it. VNTRs. We have discussed the last time, but this is very important and complete. That is why the latest DNA fingerprint highly depend on STRs. DNA fingerprinting. Okay. Then the next most important DNA marker is single nucleotide polymorphisms, SNPs. SNPs, single nucleotide polymorphisms. Here's the thing, but say with respect to genome locations, one person may have one base, say G, second person may have another base, say C. What exactly it means? For example, say DNA 1 of one organism, say this is one sequence, and this is DNA 2, this is DNA 2. At a particular location, rest is same. At a particular location, it contains G and here C. That's what exactly called single nucleotide polymorphisms. Differences among the DNAs just based on one single nucleotide. That means the name would imply single nucleotide polymorphism. Different strands That means differences in different strands is because of one single nucleotide. Say a DNA 1, a one organism contains G at a particular location. Say for example 1, 2, 3, 4, 6, 7. Okay, at 7th position. Okay, and here C is present. That's how the single nucleotide polymorphism came into existence. Here, in case of short tandem repeats, and how we have to detect the same process just like VNTRs, you have to PCR amplified, then you have to gel electrophoresis, then detection. But here, in case of SNPs, since they are that means single nucleotide polymorphisms, there is no need of gel electrophoresis. That means no gel electrophoresis. This is a very important point. No gel electrophoresis. For identification of SNPs, there is no need of gel electrophoresis. That means they save a lot of time and energy. But the rest of the DNA markers like restriction fragment length morphisms, variable number tandem repeats, and the short tandem repeats all required isolation. That means the separation by gel electrophoresis. But here there is no need of such. And what is a technique for detection? This is an important point. It is called oligonucleotide hybridization pattern. Hybridization pattern. This is very important point. Oligonucleotide hybridization pattern. It will detect the single nucleotide polymorphism. How? We'll discuss. Okay, but the screening of this oligonucleotide hybridization pattern is very important, and who will perform that? This is very latest important technique. DNA chips. We are going to discuss about this in the present class as well. DNA chips or DNA microarrays. DNA microarrays. These are the latest DNA sequencing techniques which aids in screening of single nucleotide polymorphisms, which has been detected by oligonucleotide hybridization technique. That means analysis. Okay, we'll discuss how it went. Okay, so I told you already, it's just one single nucleotide is a difference between different organisms. Take one DNA strand. Say for example, say one DNA strand. Say there is G here. Okay. This is SNP. Single nucleotide polymorphism. Then you take a synthetic oligonucleotides. Why I said synthetic? Because the probe which has been used in the single nucleotide polymorphisms is a short DNA strand which may not exceed beyond the 50 nucleotides. That means oligo beyond the 50 nucleotides, and the same will be used in mass. That means complementary base pairing with that of the SNPs. Just it is here the thing is, if there is a slightest mismatch even at the rate of one nucleotide, no hybridization will come. That means no hybridization pattern will occur. That means to get into the hybridization between the complementary sequence, that means complementary sequences between the oligonucleotide, the synthetic form and the SNP, they should be a exact, accurate complementary. And how I will tell you, single nucleotide, say this is G, C, G, C, A, C, then C, G, C, C, A. This is very important. That means standard and stable hybridization is attained. That means between the synthetic oligonucleotide with that of SNPs of the DNA, because all the bases has been matched. Perfect complementary base pairing. If there is something happens, say for example, we'll take another example. Take one more example. The same concept, but just to differentiate for better understanding. G, C, G, C, A, C, G, C, G, C, C, A. Here, just one mismatch. No hybridization. No hybridization. I told you already. In order to undertake complementary base pairing between the oligonucleotide structure, what we call synthetic oligonucleotide that of the SNPs, the SNP sequence, there should be a standard, that means accuracy, not even one mismatch. But if there is a one mismatch, no hybridization. This is very important. For example, there is a match SNPs, that means between oligonucleotides and the SNPs. Then you can expect a hybridization, a hybridized pattern. Then the same, it is followed by screening by DNA chips. We'll discuss about DNA chips here on. So it's about the single nucleotide polymorphism. There are large number of single nucleotide polymorphisms, around 3 million bases in our human genome. 3 million SNPs in human genome. This is very important point. Just imagine 3 million SNPs. It depicts the rate of occurrence of single nucleotide polymorphisms, and also the importance in DNA as a DNA marker. Okay. Then the next comes, I told you already. After that means the detection is done by the oligonucleotide hybridization pattern, whereas screening is done by DNA chips, otherwise called DNA microarrays. That means they put a single nucleotide polymorphism. So we're going to discuss about DNA chips. DNA chips, otherwise called DNA microarray, are most important and advanced DNA sequencing tool DNA chips, otherwise called DNA microarrays. It's a powerful DNA sequencing tool because of advancements in automations and miniaturizations. For example, how it works, I will tell you. Say DNA chip is nothing but, say, it's a solid membrane. The solid surface. That means nylon or glass. Then you add a fluorescently coated test DNA. Fluorescently coated test DNA for hybridization, Then hybridization will be allowed. Then you have to observe a powerful concave microscope. Microscopic examination. They are called as hybridization signals. hybridization signals. And how it ends is very important out of all the things. So this is C, G, G, A, G, 6, 7, 8, 10, G, These are oligonucleotides. That means DNA probe 8. This is target DNA. This is target DNA sequence. The process is called overlapping of hybridization oligonucleotides. And this is the entire DNA chip sequence. It's the overall schematic representation for the mechanism of DNA chips. It's simple. I don't know what it is. DNA chips, otherwise called DNA microarray, are the advanced DNA sequencing tool because of the advancements in the automations and miniaturization process. And how it works? There are two important issues here. One, the oligonucleotide probes. The oligonucleotide probes here, which are called, that's 8 in length. That means the 8 nucleotide sites, which are used in the surface. How they are used? They add to the solid surface. That means a nylon or a glass, and they become immobilized in a defined locations. Say, in a defined location they have been immobilized. That means different oligonucleotides. So it can say better, we can say oligonucleotide chip, because a large number of different means each oligonucleotide strand have different sequence. There is no match. That means a large pool of oligonucleotide strands has been added. That means immobilized, and that means in a defined locations in a solid surface, say nylon or a glass. This is the first step. After that, this is a fluorescent coated target test DNA. You allow on the nylon surface for hybridization, what we call. The process is same. Hybridization means a complementary base pairing will be done between the target DNA with that of the immobilized oligonucleotide DNA fragments, which are just 8 nucleotide size. The most important is cDNAs and synthetic oligonucleotides are used as DNA probes in DNA chips. Oligonucleotides, and they will form complementary base pairing with that of the target DNA. Then, with the help of powerful microscopic examination, we have to observe the hybridization signals. That means identifying the location of hybridizing oligonucleotides is very, very important here. Hybridizing oligonucleotides. If we identify the location with the help of this microscopic examination, we would come to know, that means we can determine the target sequence DNAs. How? This is very important. What we call overlapping of hybridization nucleotides. For example, I don't know, 8 in size. Say this is one nucleotide, this is another one, this is another one. So the first one has been eliminated in the second, and the second has been eliminated in the third, and third has been in the fourth. And the combination will give us to the will determine the target sequence. This is the process is called overlapping of oligonucleotide DNA probes. And because of this overlapping, we can identify the DNA probes. And this is far more logical and advanced DNA sequencing tool. That means oligonucleotide probes, they immobilize on the nylon membrane or a glass surface, and you add a test sample, that means the test DNA, which has been fluorescent, and allow for hybridization, that means complementary base pairing, and the microscopic exam you have to observe the hybridization signals and all overlapping of oligonucleotides, then you can determine the target sequence. It's a overlapping gist of DNA chip, otherwise called DNA microarrays. And they have lots of application. I have already discussed. One of them is it is an important tool in identifying, that means after we detect the SNPs, the screening of single nucleotide polymorphisms with that of the oligonucleotides is done by DNA chips. Apart from this, they do have other applications like, there is for in genes determining for nuclear neural system, inflammatory reactions, that means genes responsible for inflammatory reactions. At the same time, they also play an important role in total gene mapping of Escherichia coli, Saccharomyces cerevisiae, and yeast, and also play an important role in gene expression, majority of gene expression of prokaryotes, and environmental monitoring as well. That means, say, Escherichia coli, total genomic array, simply called total construction of arrays of genes of Escherichia coli has been done, and most of the genes of Saccharomyces cerevisiae and yeast has been done, and most of the gene expressions of prokaryotes has been done by these DNA arrays, and also they play an important role in environmental monitoring. That means all these are besides their role in SNPs. They actually is that an absolute technique, but they do have limitations. For example, with respect to eukaryotic genomes like human genome, there is an unavailability of complete genome array, lack of complete genome arrays. It is with respect to a particular gene array. That means you can sequence a particular gene. For example, you want a particular gene because it has some disorder or it has some connecting system like nervous system or some inflammatory system, and you will sequence with the help of DNA chips. But lack of complete genomic array of high organisms like eukaryotes, particularly human beings, is a drawback in DNA chips. But in years to come, it will be an important role. And one more important part, the recombinant tools are so advanced that we are going to get our DNA profiles. That's like ID cards we have, say the name of the college, then the name of the blood group, etc. We also may get DNA profile, our own DNA profile based on powerful automated techniques. The days will come. According to the drug design, according to the kind of DNA profile we may have, we may get our medicines. Such days are in near future. So it depicts how the recombinant DNA technology plays an important role essentially in our day-to-day life activities. Okay, it's about the DNA markers. In next class, we are going to discuss about some other molecular tools of genetic engineering. Okay, thanks for watching. I already discussed my objective is to serve for the poor and needy students, and you have to consolidate your knowledge with my lecture with a standard book and a basic book to face effectively the exams like CSR. Okay, thanks for watching.